What happens to lungs in SARS-CoV-2? There are some hidden facts behind this. You can look for these. The drastic virus, when it enters into human body, can cause several diseases through blood, through lungs, and several other organs. This is the lungs. It's a chest X-ray of lungs showing both lobes of the lungs, right and left, and the middle part is the heart. It's a endoscopic portion where you see the trachea. Then as you pass through the trachea, you reach a point where the trachea divides into right and left bronchus. And while entering each bronchus, we can see the primary bronchus is again divided into secondary and tertiary bronchus, and we can see so many holes in that. So if we enter further, finally it reaches the alveolar. And here we can see mainly the immunocompetent cells, which are the soldiers of the body who fight against this drastic virus. So, to find out the immune response in SARS-CoV-2, first we have to understand the basic immunological process going on in our body. So, first we can see what is happening in our body. So, immunocompetence is defined as the ability of the body to produce normal immune response following exposure to an antigen. The nature of the immune response depends whether the lymphocyte is a T lymphocyte or a B lymphocyte that is involved in the cell-mediated immunity or humoral immunity. That means killing by organism or any foreign particles, whether it is a bacteria or a virus, by production of antibody is the humoral immunity, whereas when the intracellular organism is perfectly killed by the T lymphocyte. Now we can see the classification of the immunocompetent cells. The soldiers are mainly classified in different types, that is T lymphocytes, B lymphocytes, natural killer cells, and there are other cells also. First we can see about the T lymphocytes and B lymphocytes in general. These are naive lymphocytes and lymphoblasts. The naive lymphocytes are mainly the resting B lymphocytes and T lymphocytes. They have not interacted with antigen and they are small lymphocytes because of small size and small cytoplasm and small amount of organelle is in it. It has a short lifespan of few weeks to months. About the lymphoblast, when the naive cell combines with an antigen, they transform into lymphoblast, which are further differentiated into effector or memory cells. And about the effector cells, they have a short lifespan of a few weeks. They are larger lymphocytes, have a large cytoplasm, larger size, and larger number of organelles in it. For example, there are effector B cells and effector T cells. The effector B cells are the antibody producing plasma cells, whereas effector T cells are the helper T cells and cytotoxic cells. And about the memory cells, they are dormant like naive cells. They transform into effector cells rapidly when there is a subsequent antigenic challenge. They have a long lifespan and provide long-term immunity. They differ from the naive cells by the presence or absence of certain surface markers like CD45RO, etc. Now we can see about the T lymphocytes in general. The T lymphocytes constitute 70 to 80% of the blood lymphocytes. They lack microvilli on their surface. They are specialized surface receptors called T cell receptors or TCR. The significance of the microvilli, because these T lymphocytes is motile, that means it can migrate from one part of the body to another part with the help of this microvilli. And about the T cell receptor, we can see in detail. It's a heterodimer like a protein chain made up of 4 encoded regions, mainly the transmembrane domain, extracellular domain, and the cytosolic tail. This extracellular part contains an alpha and beta chain containing constant and a variable region. The antigen binds to the beta region and complexes with the CD3 molecule, which is made up of epsilon delta, gamma epsilon, and zeta zeta. So, when the antigen binds to the T cell receptor, the signal will be generated and it is transmitted to the CD3 complex and lead to activation of the T cells. Therefore, when the TCR or T cell receptor is active, when both the chains complex with CD3. Therefore, it acts as the immune surveillance cells on the epithelial surfaces and form a defense against the intracellular bacteria. Now we can see the classification of the T cells. The T cells can be further classified as functions based on the functions and also based on the surface receptors. First, based on the surface antigen markers. The T cells can be identified based on the antibodies produced on the surface by the surface antigen markers or surface proteins known as cluster of differentiation protein or CD protein. There are several surface markers which can be noted from 1 to about 364, but out of these 150 CD markers have been identified yet. Some of the important surface markers of T cells include CD5, CD7, CD4, CD8, CD3, whereas a B cell is CD19, CD24, CD22, etc. Here we are mainly dealing with the surface markers of T cells. The CD4 cells, these are present in 65% of the circulating T lymphocytes. It indicates the helper function. They are produced by the macrophages and monocytes. It recognizes antigen by the MHC class II molecules. It also acts as a receptor for HIV in AIDS. Now about the CD8 receptor cells. They are suppressor on the suppressor and cytotoxic T cells. They recognize antigen by the MHC class I molecules. And about the CD3 surface markers, these are present in all T cells. They are made up of 4 polypeptide chains, as I have told you earlier. They transmit signal to the interior of the cell for an antigen binding. About the CD2, it acts as a receptor for sheep RBC. Now based on the functions. In the base of the functions, T cells can be divided into regulatory T cells and effector T cells. Regulatory T cells include the T helper cells or CD4 cells or suppressor T cells or CD8 cells. Whereas effector T cells include cytotoxic or cytolytic T cells, related hypersensitivity T cells, mixed cell lymphocyte reactive T cells. Now about the T helper cells or CD4 cells. They constitute 65% of the circulating lymphocytes. There are 2 subsets of T helper cells, mainly T helper 1 and T helper 2. The T helper 1 cells produce cytokines like interferon gamma, interleukin 2, which lead to activation of macrophages and promotion of cell-mediated immunity, and also cause destruction of the target cells and killing of the intracellular organisms. And about the T helper 2 cells, they produce interleukin 4, interleukin 5, interleukin 6, which lead to stimulation of B cells and produce immunoglobulins. And they also help in the transformation to lymphoblast and maturation to plasma cells and memory cells. As the recently detected T helper 17 cells, they are named so because they produce interleukin 17, interleukin 22, which play a major role in the inflammation and recruitment of neutrophils and play a major role in the autoimmune diseases, inflammatory bowel disease or rheumatoid arthritis. And about the suppressor T cells or CD8 cells, they constitute 35% of the circulating lymphocytes. They possess CD8 markers and MHC class I restriction. They downregulate the immune response and check overstimulation of B cells. The balanced activity of CD4 and CD8 cells results in the optimal immune response. The overactivity of T helper cells and decreased activity of suppressor T cells can lead to autoimmunity, and vice versa can cause immunodeficiency state. And about the effector T cells, these include cytotoxic or cytolytic cells. They contain CD8 surface marker and MHC class I restriction. They kill and lyse the target cells carrying new or foreign antigen like virally infected cells. And about the delayed type hypersensitivity cells, they cause type 4 hypersensitivity. It contains CD4 surface markers and they secrete different interferons like interferon gamma and cytokines. And about the mixed cell lymphocyte reactivity cells, they help in rapid proliferation of mixed cell lymphocyte reactivity. And besides, there are memory T cells of both the CD4 and CD8 T cells. They are responsible for the anamnestic immune response. Now we can pass over to B lymphocytes. These are the mediators of human immunity. That means they produce antibodies. They constitute 10 to 15% of the blood lymphocytes. The term B arises from the cytomaturation, that is, bone marrow in humans or basal fabricators in birds. They proliferate in bone marrow and in peripheral lymphoid organs like spleen and lymph nodes. Now, about the role of the mature B cells. When the antigen binds with this B cell, it gets activated and will be differentiated into plasma cells and memory cells which can produce immunoglobulins. Now we can see the difference between the T cells and B cells. The T cells originate from the bone marrow and B cells also from the bone marrow. Maturation is different for both, that is, the T cells in thymus and B cells in bone marrow. In peripheral blood, about 70 to 80% of total lymphocytes occur in the peripheral blood, whereas 10 to 15% of the total lymphocytes occur in the B cells. Now about the antigen recognition receptors. The T cell receptors are complex with CD3 in T cells, whereas B cell surface receptors contain IgM or IgD, complex with the Ig alpha or Ig beta. About the CD markers, the CD3, CD4 and CD8 for T cells, and the CD markers for B cells include CD19, 21 and 24. And there are thymic specific antigen present in the T cells, where it is absent in B cells. And T cells contains microvilla and surface, where it is absent in B cell. And surface immunoglobulin is present only in B cell, where it is absent in T cell. The rosette will be formed by the sheep RBC in T cells, where it is absent in B cells. Now about the natural killer cells. These are defined as large granular lymphocyte, constituting about 10 to 15%. And they are cytotoxic. They are part of the innate immunity and they act as the first line of defense, and they do not require prior contact with any antigen. About the site of action. They act on the virus-infected cells and tumor cells till the T cells are activated and take over the function. And they are not antibody dependent or MHC restricted. They are active in severe combined immunodeficiency diseases where mature T cells and B cells are absent. They have the surface markers CD16 and CD56. Now we can see the mechanism of NK cell mediated cytotoxicity. This NK cell have glycoproteins which can bind with the glycoproteins of the normal cells or viral infected cell, and these cells are not MHC restricted. This NK cell can differentiate the two types of cells, that is normal or abnormal cell, with the presence of two receptors, division receptors and activation receptors. The activation receptors like CD16 binds to the glycoprotein on target cells and NK cells are activated. And about the inhibitory receptors, they recognize the part of the MHC1 molecule in all nucleated cells, and the binding of the inhibitory receptor without of the abnormal cell can produce inhibitory signal and can suppress the NK cell. In virus infected cells, this MHC1 will be decreased and there will be no inhibitory signal, and there will be overactivation of the activation receptors, and this leads to production of perforins and granzymes which can create pores on the cell membrane and destroy the target cells. Besides this, there are other mechanisms also for the NK cell mediated cytotoxicity. And now you can see the difference between the NK cell and T cells. This NK cell surface markers include CD16 and CD56, whereas the T cells, as I have told you earlier, that's CD3, 4 and 8. MHC restriction is present only in T cell, it's absent in NK cell. There is no memory cell for NK cell, memory cell is present for T cell. And the T cell is a part of acquired immunity, whereas NK cell is a part of the innate immunity. And the mechanism of action is that both produce perforins and granzymes, but it is constitutive in NK cell, whereas it is inducible in T cell. Both have a cell-mediated immune response. And about the activated macrophages, the monocytes will migrate to tissues that are known as macrophages. And these macrophages are motile and move by amoeboid movement, known as wandering macrophages, and these are known as pre-macrophages. When the monocytes becomes non-motile and reside in a particular tissue, these are known as fixed macrophages. And about the secretory products. The secretory products of these macrophages include the enzymes, lysozymes, elastase, coagulase, lipase, etc. And the cytokines include interferon alpha, interferon beta, and there are also interleukin 1, interleukin 6, interleukin 12, and TNF alpha. Growth factors like colony stimulating factor, platelet derived growth factors, Other free radicals like superoxide anion, hydroxyl radicals, hydrogen peroxide, hypochlorite anion, etc. Regarding the activation of macrophages, the functional efficiency can be increased by the lymphokines, complements and interferons, and these are not antigen specific. They are larger and adhere better and spread faster on the glass, and they are more phagocytic. Some of them are known as armed macrophages because they have antigen specific cytotoxicity and can be activated by certain lymphokines or cytokines, and they are useful in anti-tumor cells and in graft rejection. Now about the role of activated macrophages. They play a major role in cytotoxicity, chronic inflammation in joints, space occupation, and excessive cytokine release can result in shock, circulatory collapse, hemorrhage, and necrosis. And the macrophages can be known by several names in different tissues. In liver, they are known as Kupffer cells, in lungs, alveolar macrophages, brain microglial cells, kidney mesangial cells, placenta, Hopfner cells, etc. And there are other cells include like dendritic cells, granulocyte cells like neutrophils, basophils, eosinophils, and other mast cells also. About the dendritic cells are named so because of the extensions like as of a neuron, and they originate from the bone marrow and they are non-phagocytic. They capture, process, and present antigen peptides to helper T cells. They carry high level of MHC class 2. They act as the messengers between the innate and adaptive immune response. And about neutrophils, they are principal phagocytes of innate immunity. The level is increased under interleukin 8, and the basophils are non-phagocytic, as eosinophils are phagocytic. And about the mast cells, they are present in skin, connective tissue, and mucosa of the respiratory and intestinal tract. And they are rich in histamine, and they have a role in allergic reactions, especially type 1 hypersensitivity. Now, as we are going through this pandemic, SARS-CoV-2 or COVID-19, you can have an idea about what the immune system doing with regard to SARS-CoV-2. As we all know, SARS-CoV-2 is a single-stranded positive sense RNA virus, which is an enveloped virus containing a nucleocapsid protein, spike protein, membrane protein, and envelope protein. It is mainly transmitted by the infected droplets in the air and also by the direct contact, also by touching the nose, eyes, and mouth, and also by the droplets on the surfaces. As soon as the virus enters in a human body, there is a receptor on the epithelial cell known as ACE2. It's angiotensin converting enzyme 2. This receptor has a higher expression in adults than in children. So, when these receptors get expressed, these receptors also present in major organs, that is liver, heart, kidneys, brain, etc. So, if this receptor get expressed, the level of these enzymes get raised on these organs and can damage these organs also. So now we can see about what is soldiers in our body doing in regard to SARS-CoV-2. As soon as the ACE receptor binds with the spike protein of the SARS-CoV-2 and the enveloped virus, the RNA is released and is transferred and is divided into multiple infected virus particles. By the translation, these virus particles, when enter an infected lung, produces interleukin 8, interleukin 1, which can lead to production of neutrophils and the other T cell activation. Simultaneously, there is toll-like receptor which gets sensed by the influence of the SARS-CoV-2 virus. It can lead to further production of interleukin 1, interleukin 6, tumor necrosis factor alpha, interferon 1. And many of these B cells becomes activated and produce virus-specific antibodies. There are also T cytotoxic cells which kills the virus-infected cells. The neutrophils also kills the virus by oxidative burst and by defense secretions. Besides, the T helper 17 cells also will be produced, which can lead to production of interleukin 17, which further leads to production of interleukin 1, interleukin 6, interleukin 8, TNF alpha, and all these lead to increased recruitment of further neutrophils and monocytes. This interleukin 17 play a major role in severe inflammatory respiratory diseases. In mucosal immune response, interleukin 22, interleukin 17 also play a major role, and this interleukin 22 can cause life-threatening edema and lungs become enriched with mucus and fibrin, and this lead to pneumonia and ARDS, that is acute respiratory distress syndrome. And therefore, the chronic stimulation of these T cells lead to cytokine storm, that is excessive cytokines. All these factors will be produced excessively in the body, and this can be seen in the form of coagulopathy, multiple organ dysfunction, raised creatinine. Just a typical of cytokine storm, and there is become the T cells or CD8 cells get exhausted. And therefore, the chronic stimulation of the T cells can lead to cytokine storm and the T cell exhaustion, weakens the body defenses and put the patient in a dangerous condition. All these can lead to decreased effector function and overexpression of their inhibitory receptors like CD279 that act as a mature T cell for apoptosis, lead to suppression of the immune system by affecting the T cells, B cells and NK cells. This leads to lymphopenia in COVID-19 patients. So to conclude, the innate immunity by neutrophils and pro-inflammatory cytokines lead to cytokine storm, and the body trying to limit the infection and overcome the virus, lung injury, tissue damage and life-threatening edema by mucins and fibrin occurs, and this results in the ARDS and death of the patient. Thank you.